My name is Jessica. I'm a PhD student in MR Physics at the University of Basel, and I will tell you something about myelin water fraction imaging. Now, that might sound like a complicated word, but let's start simple. Your brain, and in fact all of your nerves, are made up of cells called neurons, which look like this. They are responsible for carrying orders from the brain to every part in the body, and they do this by electrical signals. However, neurons can't do this job on their own. There are a lot of helper cells called glia cells. Glia means glue in Greek, which comes from the fact that people originally thought that these cells are only there to keep everything in place. This could not be farther from the truth. Glia cells do all sorts of stuff. For example, some of them snug really tightly around the neurons in multiple layers to isolate them against each other, similar to the coating around an electrical wire. They're also crucial in speeding up signal transmission. Part of our brain, called white matter, contains an enormous amount of glia cells. Here the coating is called myelin, and it is very important for the flawless function of our brain. There are a ton of diseases which affect myelin. For example, you may have heard of a disease called multiple sclerosis. In this disease, the body's immune system accidentally attacks the myelin. So the glia cells which make up the myelin are slowly dying, leaving the neurons alone and exposed. Patients with multiple sclerosis usually have problems with vision, speech, and mobility. So as you might imagine, we want to understand the progression of these diseases in order to develop better treatment. One way to detect the decrease of myelin is by measuring the percentage of it inside the brain. What we can use to our great advantage is that the water, which is trapped between the myelin layers, behaves differently than the surrounding water when put inside of a magnetic field. When we excite these different water pools with radio waves while they are inside of the magnetic field and measure their reaction, we can see a difference. This difference is not directly visible. The data we acquire is a mashup of the contributions from myelin and the surrounding tissue. By application of a special model, one can calculate the ratio of these contributions and therefore the so-called myelin water fraction, which is directly dependent on the percentage of myelin inside the brain. This is how a typical map of the myelin water fraction inside of a healthy brain may look like. The bluer a region is, the higher the percentage of myelin. You can see the similarities to the drawing of white matter I showed you earlier. Now, obtaining useful data is not always as simple as it sounds. There are a lot of different effects taking place inside of a magnetic field, and a lot of them are impedimental and make it such that the data can't be described by a simple model. This makes it a lot harder to evaluate the data correctly. Most of these effects scale with the strength of the applied magnetic field, and while strong magnetic fields have a lot of advantages in other cases, they hinder the evaluation in this one. In my work, I applied the described method at lower magnetic field strength than hospitals usually use to keep these effects as weak as possible. Here the used model is more accurate, which could lead to a more reliable evaluation of the myelin water fraction, making it more suited for clinical use.